Welcome everybody to this H3D Foundation webinar. I'm Nicola Elliott-Wang, the Program Coordinator at H3D Foundation, and it's my great honor to host these sessions. And today we're super excited to have Dr. Walid Houry from the University of Toronto with us. And for our GCADA flagship, TB flagship members, this one is especially for you, but we know that it's going to resonate across the board of our network. We're really excited. So I'm going to start by introducing Dr. Houry. So Dr. Walid Houry is a professor in the Department of Biochemistry, Temerty Faculty of Medicine, University of Toronto. His lab studies protein homeostasis, focusing on how molecular chaperones and ATP-dependent proteases maintain cellular proteostasis, and how these systems can be targeted for drug development. His group combines structural, biophysical, biochemical, and proteomic approaches to address how protein quality control is maintained in the cell. His research has been recognized nationally and internationally, including the Tokyo Biochemical Research Foundation Award, the National Research Foundation of South Africa Visiting Scientist Award, Premier's Research Excellence Award, and the CIHR New Investigator Award. Dr. Houry currently serves as the president of the Canadian Society for Molecular Biosciences, scientific officer and vice chair of the Banting Discovery Foundation, and chair of the American Society for Biochemistry and Molecular Biology Publications Committee. He was recently appointed as associate dean in the Faculty of Medicine at the University of Toronto. So today he's here to talk about ClpXP proteolytic complex, from basic principles to drug discovery. And Walid, we're really thrilled to have you. Thank you so much for carving out the time. I don't know how you've done that, because you're an extremely busy person, as we can tell. But I'm going to take this moment to welcome you, to say thank you, and to remind everyone that we'd love to have an exciting Q&A at the end. Don't leave that to me, please. Ask your questions yourselves. You can pop them in the chat, or write them down, and I'll unmute you at the end of the session. So welcome everyone, and over to you, Walid. Thank you so much.
Well, thank you very much, Nicola. This is really a pleasure to be here. I want to thank also the foundation, the H3D Foundation, for putting my name forward to present this seminar. Africa has a special place in my heart, especially South Africa. I've visited there once, and it's just an amazing place as well. And I'm born actually in Africa, and that's why I always feel some attachment to that region. So my lab works on the general question of protein homeostasis in the cell. So basically, how do proteins fold, how do they assemble, and how do they get degraded? So we do a lot of work, just the basic biology, trying to understand how this happens, how the structures look like, especially in studying the molecular chaperone complexes, as well as these ATP-dependent proteases. Now, for this seminar today, talking to Nicola, I think the interest is more on the drug discovery angle, I was told. So this is something my lab is also interested in. So basically, we are interested in targeting these chaperones and proteases to develop new anti-cancers or antibiotics. And so today I will describe to you our efforts in targeting this proteolytic complex, which is called the ClpXP. I hope everyone is able to see the slides. So basically, this has been an ongoing project for quite some time. There's many people in the group that have been involved in this. I just want to acknowledge them here on the left. Recently, it has been Mark, Trevor, Mazen, Emily, and Chen have been involved in this very heavily. And we've had many collaborators across the University of Toronto, especially Rob Beatty, who's been really my MedCamp collaborator for a long time. So what is ClpXP? So I will concentrate on the ClpXP in the mitochondria. So this proteolytic complex also exists in bacterial systems as well, but for the presentation today, I will concentrate on it from the mitochondrial perspective. So it is an ATP-dependent protease. It's present in the mitochondrial matrix, and it looks something like this. It has a cap, which is called the ClpX, and the core that's called the ClpP. So for those who are familiar with the proteasome, it looks very similar to the proteasome, although it's simpler in its constitutive subunit assemblies. But it is considered to be, maybe you can think of it as the equivalent of the proteasome, but in the matrix of the mitochondria. There are actually other ATP-dependent proteases in the mitochondrial matrix, including LON and the MAAA. So, and we know from studies in my group as well as many other groups that the ClpXP is involved in many different pathways that are impacted by the mitochondrial translation, amino acid biosynthesis, energy metabolism, UPR stress signaling as well. So the mitochondria undergoes a stress signaling pathway, which is called the unfolded protein response, but is a mitochondrial version, not the ER version. I think most people are familiar with the ER version. So based on a lot of proteomic studies that we and others have done, we have basically a hit list of proteins that are impacted by the ClpXP. So that might be targeted for degradation. But again, this is based on proteomic studies. So some of them might be false positives, others might be false negatives. So, but basically just to see that it has impacts across many different complexes in the mitochondria. So somehow it impacts especially complex 1 from the respiratory chain complexes, as well as complex 2. That we've verified. It impacts translation of ribosomes by ribosomes. It impacts chaperones within the mitochondria, as well as things like fatty acid metabolism, pyruvate metabolism, and amino acid metabolism. So it has a very varied effect on the mitochondrial proteostasis by targeting many of the mitochondrial proteins. So from the cancer angle, it turns out that this system is actually upregulated, heavily upregulated in many, many cancers. So whether it's colon, liver, prostate, or uterine cancers, and this is showing an immunohistochemistry of tissues from cancer patients, showing that the ClpP is upregulated heavily in the tumor. And this is really seen here. If you look at this slide here, this is basically what checking the levels of the ClpP in the tumor versus in the surrounding normal tissue. And you can see that the ClpP, the protease part, is highly upregulated. Unlike, for example, LON, which actually goes down in a tumor compared to the ClpP, compared to the normal tissue. So how does the ClpP look like? So initially, I'll describe the P, which you can think of as being the core particle of the proteolytic complex. So it's a 14-mer. It's made up of 14 subunits. And one subunit looks something like this. So it has a head domain and a handle domain. It looks like basically a hammer. So the handle domain would be the handle of that hammer, and the head domain would be the head of that hammer. And the catalytic triad, so it's a serine protease. So it has a SEER, HIS, ASP catalytic triad. And the catalytic triad sits here. And you have 7 of these coming together to form a ring, and then another 7 form another ring. And the 2 rings come together to form the cylinder, and this is shown on the right here. So the cylinder is basically a 14-mer, and you have 14 subunits basically encompassing a catalytic chamber, and the catalytic chamber is inside the ClpP. And the catalytic chamber basically now consists of contains 14 catalytic sites. So a substrate has to enter through what we call the axial pore. And this is shown, this is a side view, this is a top view. So a protein has to enter here, enter into the catalytic chamber. It gets degraded into very small peptides of around 10 residues, and then these peptides are released. So we and also others have solved the structures of the ClpXP from many different species. I'm showing you one of those structures. Again, so the ClpX would be the cap, and this is the orange part here. ClpP would be the core, which is the green part here. And the cap comes and docks onto the ClpP cylinder. And it can dock on one or both sides, actually, of the ClpP. And the cap will then recognize the protein, unfold the protein, pushes it in and into the cylinder for degradation. So let me show you this again in the next slide here. This is just schematically of how we think the system works. This is based on a lot of biochemical and structural work that has been done across the years. But basically, you have the blue folded protein. It will be unfolded by the cap, the orange part. And this unfolding step requires energy, so it's ATP-dependent step. The blue protein will be unfolded, pushed in and into the green ClpP cylinder. And then the ClpP will act on the protein processively to degrade it into small peptides. These peptides are released, and then the cycle will repeat. And we call the ClpXP complex as a processive protease, meaning that if you give it something that's 100 residues, you get back 10 residue peptides. If you give it something that's 1,000 residues, you get back 10 residue peptides. So it is not like trypsin or chymotrypsin where it cleaves slowly and you get intermediates. We don't see those intermediates. The protein stays in the catalytic chamber until it's degraded into these 10 residue peptides. So in targeting ClpP, so basically it's a protease, so you can inhibit it, and that's something we worked hard on. But it turns out that inhibiting the protease turns out to be difficult. You can inhibit it by anything, even by adding PMSF, you can inhibit the protease. But finding a specific inhibitor that only inhibits ClpP and nothing else in the cell has been very problematic. We could not, and others groups and actually companies worked on this, and no one has an inhibitor that only targets ClpP and nothing else, because it's such a broad category. It's a serine protease. So my lab then started thinking of another strategy. And the strategy that we now work on is what we call activation or dysregulation of the ClpP. So let me explain this. So the ClpP, again, this is the ClpP cylinder by itself. And in red are the pockets. These are hydrophobic pockets where the cap comes and docks. So the ClpX comes and docks at these pockets. So we identified compounds that actually bind at these hydrophobic pockets. So by binding at these hydrophobic pockets, the compounds prevent the interaction between the cap and the cylinder. So the cap cannot dock anymore. But more importantly is that what the compounds do is, what also the cap would do, is that open up that entrance pore. So if you can see on the left-hand side here, the entrance pore is actually closed because there are these axial loops that are floppy and they hinder entrance into the catalytic chamber. But when the compounds bind, these axial loops are actually ordered. They are moved away from the entrance pore. And then what happens, our proteins start going into the cylinder unspecifically, and then they get degraded. So there's no selectivity anymore to what you are degrading in the presence of these compounds. So now we have a cylinder with a very open pore that's eating things up inside the mitochondria unspecifically, and eventually you're causing stress to the mitochondria because you're degrading the proteins from within, and this eventually leads to cell death. So this is the trick that we've developed, and this is called ClpP activation or dysregulation. And the compounds are called activators, although the more proper naming would be dysregulation, because basically now you're degrading things unspecifically rather than selecting them through the cap. So the cap is what selects the proteins to be degraded, but now we don't have the cap. We just have an open pore, and anything that can go in will go in and get cleaved and degraded. So I will describe to you two types of compounds that we worked on. The first one is a natural product that was initially described by Heike Brotz-Osterheld when she was at Bayer. And these are the acyl-depsy peptides, so shown here. And these are basically cyclical peptides. And these peptides, these ADAPs, we call them acyl-depsy peptides, the ADAPs, they were identified as small molecules that were secreted by streptomyces. And actually, streptomyces are the ones that discovered this trick, not us. So basically, it was a bacteria doing this trick that Heike identified and showed that, yes, these compounds do the trick that I just mentioned. And then what I will describe to you is more potent compounds that we worked on. These are these types based on the ONC201. These are the imipridone-based compounds. So this is the outline of what I want to describe today, basically our development of the ADAPs and then the imipridones, which we call the TR compounds. And then I'll touch briefly on the consequences of what happens to ClpP and the effect on the animal, on the cell, and on the protein. So we'll start with the ADAPs because this is how we started the project, basically. So the project was initially more bacterial oriented. So we were developing these ADAPs, we were synthesizing them, many different types of modifications to the original ADAP. So this is like ADAP 28, ADAP 41. So we generated like 100 plus analogs of these compounds. And initially we were interested in targeting the bacterial ClpP, and we were counter screening against the human ClpP. And it turned out, just by accident, many of the ADAPs that we made were pretty good against the human ClpP. And this is a quantitative number that we use, an RD number, relative degradation index number. And the closer it is to 1, the better is the compound at activating the human ClpP. So it turns out, just by accident, that many compounds were activating human ClpP. And my postdoc then wanted to push this further. I thought this is going to be problematic because these compounds would have to cross in the mammalian cell, they would have to cross the plasma membrane, the outer membrane of the mitochondria, the inner membrane of the mitochondria to reach the matrix of the mitochondria. So I did not think they would work on the cell, on the mammalian cell. But we persisted, and actually, as I will show you, actually the compounds do reach the matrix of the mitochondria. So initially, I'm going to show you that the compounds work in a test tube. So this is just using purified components, purify the human ClpP, purify the human ClpX. And we use a model protein called casein as our substrate. So as you can see, under the conditions of this experiment, when we add ATP, we cannot really degrade the casein very well in the absence of the compounds. But if we add some ADAP at low concentration, okay, the degradation also does not happen, but at a bit higher concentration at 2 micromolar versus 0.1 micromolar, we can see that the casein gets degraded. And so basically, then we think the compounds are working, at least using purified proteins. And based on a titration experiment, the KD for the compound binding to ClpP is around 0.5 micromolar. So this is just titrating ADAP and checking activity of the protease. The important experiment is the following. So we had generated, we have, let's say, HEC293 cells, but we have many different cancer cell types as well. And we generated cells that are deleted of the ClpP. So in a lab, we can delete ClpP and the cells survive. So this is the important experiment whereby when we add the compound to the wild type cells, so this is the closed circles here, the black curve, then we see that we can kill the cell. So this is a cell viability, and the IC50 is again in the sub-micromolar range. However, if we use cells that are deleted of ClpP, then we cannot kill those cells until, of course, you go to very high concentrations and becomes unspecific. So this was very critical because it told us that if there's no ClpP, the compounds have no effect on the cell. Because basically, it tells us that the compounds are on target. They need to interact with the ClpP. They need to activate ClpP to be able to kill the cell. So to show also that these compounds are on target, because we have many different analogs of the ADAPs that we generated, we can show that the IC50 that we obtain correlates nicely with the KD on the purified protein. So the IC50 on cells correlates nicely with the KD on the protein. And we get a nice correlation curve. We also show that for different types of cell lines, different HEC, HeLAs, U2S, SH, SY, 5Ys, the IC50s are all in the sub-micromolar range, although the ClpP levels might vary. So this is like in HECs, ClpP levels are much higher compared to U2S, say, or SH, or the SH, SY, 5Y. But the ADAPs induce cytotoxicity in these several different types of cell lines. So what are the compounds doing? So we can show that the compounds are inducing actually apoptosis. So this is just wild type cells treated with the compound, with an ADAP compound, and or DMSO, or the ClpP knockout cells treated with ADAP or DMSO. And as you can see, this is a tunnel assay showing the existence of apoptosis and DNA fragmentation. We can see that in the presence of the ADAP, upon addition of the ADAP to the wild type cells, we see apoptosis happening. but not if we have the MSO added to wild-type cells, or ADAP added to the ClpP knockout cells, or the MSO added to the ClpP knockout cells. And this is quantified here. So again, telling us that we are inducing apoptosis under these conditions. So another thing we checked, checked the effect on the pro-apoptotic markers and the anti-apoptotic markers. So this is just an example. Again, if you look at the wild-type columns here, so when we add the ADAP, this is a 24-hour treatment. Actually, the ClpX is degraded. So the cap itself is degraded. The MCL1, which is an anti-apoptotic marker, it goes down. PUMA, which is pro-apoptotic, goes up. Again, telling us that we are activating, which I'll show you later, is a caspase-dependent pathway. So these are the caspases that we Western blotted for. So this is the caspase 9, and we can see cleavage. You can get a cleavage of caspase 9. You see the P37 and P35 bands for the caspase 9. For the caspase 3, you see the P19, P17 band. So again, this is the intrinsic caspase-dependent pathway being activated in the wild type. In the ClpP knockout, we see no such activation, whether we have the drug or we don't have the drug. So again, if you look down, we don't get the cleavage of the caspase 9, and we have no cleavage for the caspase 3. And finally, the caspase 8, which actually is more of an extrinsic caspase that's activated, that gets activated upon the presence of an extrinsic signal. We don't see anything happening to that. So again, telling us that the activation of ClpP leads to an intrinsic caspase-dependent activation pathway. So just to show you what happens to the mitochondria, this is a 3D SIM image of wild-type cells and the ClpP knockout cells. This is upon addition of the MSO versus the ADAP. And you can see that after 72 hours, the mitochondrial network in red is maintained in the DMSO-treated cells, but basically there's huge integration of that network upon addition of the ADAP. In the ClpP knockout cells, that network is really not affected. So again, the red would be the mitochondria, and the mitochondrial network is not affected upon addition of the compound. Again, telling us that the compound is on target. Subsequently, we solve the crystal structure of the ClpP with the compound. So this is the ADAP, and it sits where it's supposed to sit, basically at these hydrophobic pockets. So these hydrophobic pockets are actually at the interface between two subunits. So this is a top view showing you the top of the ClpP, and the ADAPs are here in white, cartoon presentation here. And they sit at the interface between two subunits as expected. So these are the hydrophobic pockets that where ClpX would sit as well. And this would be the apostate, and you can see that the entrance pore in the apostate is smaller compared to the entrance pore in the ADAP bound state. So this is a zoom in into that binding pocket. So this is the apo, and so this would be one subunit, let's say in red, and the other subunit in blue. And then when the ADAP comes, it sits at the interface between these two subunits. And therefore, the ADAP seems to act as a wedge to open up that entrance pore, and I'll explain more what's happening. So we've worked on these ADAPs for quite some time, trying to push them further. So basically, as I said, we are in the sub-micromolar range, but we could not push them down to much more in terms of KD or IC50. So we wanted to reach more of a nanomolar range. So that's when we started using another chemical scaffold, which we call the TR compounds. So these TR compounds are based on a parent compound called ONCT01. And this compound was initially discovered in a high-throughput screen for inducers of TRAIL, which is a cytokine, and also leads to apoptosis. Initially, this compound, the ONCT01, was thought to be a dopamine receptor antagonist. And actually, it was already shown to be good against multiple cancers, and had the right drug-like properties. It's a very simple compound. We can make this in less than a week. The ADAPs, we need like a month to make. The student takes a month to make an ADAP analog. So this is very nice, has drug-like properties. and it penetrates the blood-brain barrier. And actually, there were clinical trials using this compound already, although, as I will show you, it does not target the dopamine receptor, and actually it targets ClpP. So we have a collaboration with Madeira Therapeutics, and they generated many, many analogs of the ONC201 by modifying different parts of the molecule, either the eastern fragment, the western fragment, or this part here, which is called imipridone. So this is an imipridone moiety. So we initially checked these different analogs against a panel of cancer cells, like we used the NCI 60 cell lines. So these are 60 different cell lines that you can screen against. We used TR57 and TR107, and all these cell lines were affected in their growth upon addition of these compounds, and a couple of cell lines were even killed using these compounds. So they are very effective against multiple cell lines, cancer cell lines. To show you the IC50 of these compounds, so this is against MB231s, which is a triple negative breast cancer cell line. And now we are in the nanomolar range. So the unit here is nanomolar. And you can see that we are, like, TR27 is 90 nanomolar, but TR65 is 10 nanomolar. So we are much, much better than the original ONC201, which is 1.7 micromolar. Importantly, again, if we knock out ClpP from the MB231s, then the compounds cannot kill the cells. So we don't get cell killing. If we get a ClpX knockout, the compounds still work. So again, telling us that the compounds are on target. And we get similar results, for example, using another cell line here. I'm showing you just the HEC, which is our workhorse cell line. It's these cells we like. They're easy to work with. And again, we have an IC50 in the nanomolar range, while the ONC201 is in, let's say, 0.75 micromolar. Again, if you knock out ClpP, the compound do not work because the target is not there. If you knock out ClpX, the compounds still work because the ClpP is still there. And then this is the KD on the purified protein. And again, the KD is in the nanomolar range. So around 20 to 30 nanomolar, say. And the KD for the ONC201 is in the micromolar range, 3.5 or so micromolar. So in terms of what we've done, what Madeira was doing in terms of optimizing these compounds, we found that on the eastern end, it's good to have a nitrile group or an alkyne that helped in enhancing the activity. In terms of the imipridone moiety itself, we can simplify that moiety by making a pyrimidine dione, and for example, for this compound, or pyrimidone for this compound here. On the western end, it seemed adding a chlorine or a bromine, something a bit electronegative, or actually even a fluorine, I'm not showing it here, that helps as well. And that helps in reducing the KD from, let's say, the 3.5 micromolar to the 20 to 30 nanomolar. So this is really a huge improvement. And we also solved the structure of these compounds with the ClpP. So this is shown, again, they bind where they are supposed to bind at these hydrophobic pockets. So this is, again, the top view, and this would be the enlarged entrance pore for the ClpP. So if we zoom into the binding pocket itself, you can see why these compounds are really potent. This is the binding pocket, and the binding pocket itself can be divided into smaller parts, right? So the imipridone moiety sits flat in this binding pocket, and then on the eastern end, you have a sub-pocket which is occupied by the eastern fragment of the molecule of the compound, and on the western end, you have also a sub-pocket which is occupied by the western fragment of the molecule. So we are getting really very beautiful surface complementarity of the compound sitting in this binding pocket. And that's why we think we are pushing the KD to a nanomolar range. So this is just another view. If you look here at this slide, at this panel here, you'll see that the compound, against it, as I indicated, you have the flat part, which is the imipridone part, and then you have the eastern fragment going in, the western fragment going in. And we think there's a bit more space down here that we could try to occupy, maybe to push to picomolar range. And this is something that I'm trying to advocate for with my students here. We'll see if we can go from nanomolar to picomolar. Although, I mean, in terms of a drug-like molecule, nanomolar is pretty potent already. And to show you the difference between the acyl deoxypeptides and the TR compounds. So this is an overlay of the two structures. And as you can see, again, the orange is the TR compound that sits very nicely in this pocket. And the purple is the acyl deoxypeptide. So the cyclical peptide part is the part here. And as you can see, it doesn't seem to make too many contacts. It is very difficult to synthesize, and it's not helping in terms of enhancing affinity. The acyl deoxypeptide does have a phenyl moiety here that sits in the eastern pocket, but the western pocket is not properly occupied because it has an alkyl chain. And even we tried to play with that chain to force it to go in here, but that did not help very much because of how the molecule is organized. And we think that's the reason why the acyl deoxypeptides, we could not push them further than a sub-micromolar range. So what are the consequences of ClpP activation? So I'll show you some of the animal studies that were done. This is TR107 and an MB231 mouse xenograft model. And this is measuring tumor volume at day 26. This is the control group in black. And these are group 2 and 3 are the compound treated groups. And you can see that the tumor volume is reduced. And this is as a function of time, the reduction in that tumor volume. In terms of survival of the animals, again, the black is the control group. So the animals die off, so the animals die earlier than the compound treated group, which is the red and the blue. And this is days versus survival. Finally, since the compound is really hitting the mitochondrial ClpP, and therefore we should be affecting mitochondrial mass, you can measure a mitochondrial copy number, or DNA mitochondrial copy number. And this would be, again, the control group. And then this is the compound treated group. And you can see that we are reducing mitochondrial DNA copy number, which is what we would expect if the mitochondria is being affected upon compound treatment. So what is happening on the cell level? So in this case, we are interested to know what are the proteins that are being targeted by the activated ClpP. So to do that, we carried out an experiment, which is a mass spec analysis of the N-termini of proteins using a high-chain type assay. So this is a mass spec approach that allows you to identify the N-terminus of a given protein. So if you want to think of it, basically, when ClpP attacks a protein and it's going to cleave it, you're going to generate a new N-terminus. And we were interested to know what that N-terminus is and what protein that is being attacked. So using this N-terminum analysis, we are able to identify what is the protein that's being attacked by the activated ClpP and where the cleavage is happening upon activation of the ClpP. So this is very important information for us because now we are trying to understand what is the pathways that are being activated and leading to cell death. However, we have a couple of very interesting observations. The first one is maybe what we would expect. We get a lot of mitochondrial proteins that are being cleaved by activated ClpP. And upon cleavage, their levels go down. So this is fine. This is what we would expect. But what we did not expect, we get also cytoplasmic and nuclear proteins that are being cleaved. And upon cleavage, these guys are stabilized. They don't disappear as a function of time. So this phenomena, we're not sure how to explain, whether it is a direct or an indirect effect. So basically, you can think of maybe because we are activating ClpP, there's a lot of stress. Maybe some ClpP leaks out and start cleaving things in the cytoplasm and the nucleus. Or there's something else that's happening. There's a signal from the mitochondria, and other proteins start cleaving the cytoplasmic and nuclear proteins in the cytoplasm and the nucleus. So maybe the proteasome, maybe other proteases get involved and start cleaving, just as a general stress response. So currently, we're not sure if there's, again, a leakage of the ClpP outside to the outside of the mitochondria because of the stress we're exerting on the mitochondria, or there's something else that's being triggered in the cytoplasm to start cleaving proteins as well. So this is something we're working on at this stage. So the other thing that we can look at is, because we know what are those sequences so that are being cleaved by activated ClpP. So we can then look at those sequences and analyze them. And what we see is that there's not necessarily an enrichment in a motif that's being attacked by the ClpP, but actually ClpP seems, the activated ClpP seems to cleave mostly at loop regions, so the random call regions. And this is what you would expect, because basically you have now a cylinder with an open pore. So if you have a long loop within a protein, that loop will go into the cylinder and get cleaved. Because that's the most likely scenario. We don't think, let's say, a beta sheet or a helix would be the first one to go into the cylinder. But maybe a floppy loop will be the first one to go into the cylinder and get cleaved. And then this starts to slowly unravel the protein target, the substrate, basically. So you're unraveling it by start cleaving it slowly, one step at a time. Maybe the first cleavage event happens at an unstructured loop region. And this is what we see from analyzing all those sequences that we get from the mass spec. So just to show you some of the proteins that are being cleaved by activated ClpP, this is upon treatment with TR107. We see that the cap get cleaved, so the ClpX itself. SDHA, which is the complex 2. TFAM, which is the main regulator of transcription in the mitochondria. TAFM, which is an elongation factor. So all of these get cleaved by the activated ClpP. Others, UNDAF S3 is another one. Basically, this is a complex 1 subunit. Things that we are personally interested in chaperones. So we see something like HSP60, which is a very stable chaperone in the mitochondria, is getting cleaved by the activated ClpP. So this is very interesting to us. So the HSP60, it gets cleaved whether we are using ONC201 or TR107. So again, meaning that there's really no specificity. As long as there's some floppy loop that can be attacked by the ClpP, it will be attacked by the activated ClpP. So on the protein level, so what's happening to the ClpP itself? So let me explain this schematically. We've done a lot of structural work as well as biochemical work to try to understand how ClpP gets, quote unquote, activated. So as I mentioned earlier in my presentation, is that at the entrance pore, there are these loops, which are unstructured. And basically, these loops are unstructured, and they prevent entry into the catalytic chamber by acting sort of what's called an entropic barrier, because you have floppy loops that are hindering entrance. And this is shown schematically here. So these loops are floppy, and they are just moving around and hindering entrance. And there's basically an interaction network at the entrance pore, mostly ionic type interaction network. So when the compound comes, like the ADAP or the TR, it comes and basically wedges itself between two subunits. So these compounds are acting like a wedge between two subunits. And when they do that, then there's a reorganization of that interaction network around the ClpP entrance pore. And here I'm showing you some of the details of what happens. So there's extra interactions that now happen between the loop region and the body of the ClpP itself. And what this leads to is that basically the loops become structured, and then they are retracted away from the entrance pore. And that's how we open up that entrance pore, because we are retracting away those loops by structuring them through these interactions. These interactions that we identified do not exist in the apostate. So because in the apostate, let's say the interactions are mostly between, let's say, this orange helix and the blue helix. But then in the compound bound state, these interactions are lost, and we form new interactions between the loop region and the blue helix. And this is really based on a lot of work, including structure, NMR, and others. So just to give you an overview of what we think is happening based on a lot of this type of analysis, is that in the ClpP apostate, we have these unstructured axial loops at the entrance pore. But also in the equatorial region, in the middle of the ClpP cylinder, there are these helices that also are not very well structured, so they are floppy as well. And interestingly, the active site sits here at the top of these helices that tend to be a bit floppy. So this means in the apostate, the cylinder cannot take in proteins because of the axial loops being floppy. And the equatorial region is floppy as a result. Then you have the catalytic triad, which sits here basically. The CIHR catalytic triad is not properly structured. So basically, in the apostate, we think the cylinder is really not active per se. But then when we add the compounds, then there's structuring of the axial loops, and there's stabilization of the equatorial region. And therefore, now the catalytic triad is properly oriented and is ready for catalysis. So then we have a range of states that the ClpP can exist in, and completely apostate, which is not very active state, and then a more compound-bound state, which is more a rigidified state, and the protein is in an active conformation. And this binding of the compound mimics the binding of the cap. So the cap does the same thing. When the ClpX comes and caps the cylinder, it's doing a similar thing. It's stabilizing the axial loops as well as equatorial region in an allosteric fashion. And therefore, we have a range of activation for this ClpP cylinder from a non-activated state to a fully activated state. A fully activated state would have a more open pore and a narrower equatorial region. So if you want to think of it, it's basically then we have ClpP can eat more proteins, but becomes thinner. Like that's we all dream of that, of course, yeah. So with this in conclusion, then I hope to have shown you that the dysregulation of mitochondrial ClpP is a novel approach for the development of new anti-cancer agents, but it also leads to very interesting biochemistry. And actually, in August last year, 2025, actually, the FDA approved the ONC201. Now it's known as MODAI. So for patients with a special type of cancer, it's the H3K27M mutant diffuse midline glioma, DMG. And it marked the first ever really FDA-approved treatment for this devastating disease. So this disease had no treatments before, and now this is the first, at least, treatment for it using the ONC201, which is the less potent version, and that's why we have high hopes for the more potent version. And we were the first to propose that activation of the ClpP could be used to target cancer cells. So with this, I will thank you for your time and thank the group. So this is the group just this summer. And currently, the people involved in the project are Trevor, who's a postdoctoral fellow, Mazen, an amazing graduate student. Elisa is the technician who's been on this project for a long time. Mariam in the background here, and Emily are all what we call ClpP people in the lab. So I think I'm on time, and so I thank you for being here, and I'm happy to take any questions.
Excellently on time. Thank you so much, Walid. Thank you for that amazing presentation. I'm going to suggest that people raise their hands to get in the queue to ask questions. You will be able to unmute yourselves and even come onto camera if you want. I know there's a question in the chat. Is it from Conrad? Would you like to ask your question in real time?
Should I stop sharing, Nicola?
Yeah, sure. You can, unless you want to go back to refer to your slides, which is also possible.
Okay, let me stop for now. Maybe if I can see some of the audience, or maybe not.
Yeah, feel free to come onto camera, guys, if you wish. Thank you. Over to you, Conrad, and everyone else raise your hand so that you can form a queue. Thank you.
Hi, Walid. Thank you so much for the good presentation. We are really interested in some of this ClpP work, and it's wonderful to see the work that you've been doing so far. I just had a question regarding the activated ClpP. The protein substrates that you now see that are degraded by the ClpP, the activated ones, are they also degraded into these 10 amino acid chunks as you see for the normal ClpXP complexes?
Okay, so the question is whether the activated ClpP is degrading the same protease as ClpXP, I guess. That's the question?
The chunks that they are also degraded into. As you explained, for the ClpXP, you've got this 10 amino acid peptides that they degrade into, but for the ClpP by itself, does it also do the same type of degradation?
Oh, okay, sorry. So you're asking whether the activated ClpP is processive. Then, yeah, so the activated ClpP is not necessarily processive. It's not processive because when we see intermediates upon activation of the ClpP, so it's not a processive protease anymore. Yeah, that's a very good observation. Yeah, because it's cleaving slowly, and the protein can go in, gets clipped, and then leaves again because there's no cap to keep it in. The cap is the one that keeps pushing things in and not allowing them to leave unless they are small peptides. Very good question, Conrad.
Okay, thanks, Conrad. Mokatli, Morake, if you'd like to unmute yourself and go ahead. Let's see. Oh, background noise. Okay, let me give it a go. My question is, in bacteria, what were the values? So this is in the chat, Walid. What were the values, and could these be explored as back protacs, given that some recent binders are also macrocyclic compounds? Also, for cancer applications, could these compounds be developed into protacs for protein degradation?
Yeah, actually, we are working on that. Again, very good question. We are working on that. So basically, what we are doing is, because we have all the structures, so we have a linker now attached to these compounds, and with this linker, we can attach a corresponding compound to target more specifically. So rather than basically degrading anything and everything in the mitochondria, we want to be a bit cleaner. So basically using a PROTAC type approach, we can link the TR compounds to another compound for a protein of interest. Let's say if you want to go after, I don't know, the DHFR, so you can link it to the compound that targets DHFR, or to any of the respiratory chains. So there's many compounds that target the respiratory chain. So yeah, we are doing those tricks currently, actually. So we're working on that. Again, very good question.
Okay, Adrian, go for it, and then I'll come to you, Mahmoud.
Oh, Adrian.
No, you're muted.
Sorry,
I can't hear you. Let's try again. Oh, you don't look muted.
We cannot hear you.
Okay, you're gonna type. That is odd. I hope. Just try one more time, because now there you go.
Can you hear me now?
Yes, we can hear you now. Yeah.
Can you hear me now?
Yeah.
Oh, excellent. Yes.
Oh, great. Sorry, my laptop took a fall the other day, and so it's messing around now, which is also I'm on an external screen, which is why I don't look like I'm looking at you. But Walid, thank you so much for a lovely talk, as usual. And I just wanted to ask, I'm interested in the ClpX. Okay, so I noticed that when you did the initial in vitro degradation assays, you included the ClpX in the assay. So for these compounds that target the ClpP, is the ClpX required for these in vitro degradation assays? And then what do you think is going on with the degradation of ClpX in the cells? So how much of the effectiveness of these compounds do you think is due to the loss of ClpX, or do you think that that's got nothing to do with, with the outcome?
Yeah, no. So always good to see you, Adrian. First of all, and the compounds are have a much higher affinity to the ClpP than the ClpX. So basically, they are just displacing the ClpX. So the presence of the ClpX is not needed for these assays at all. Okay. And the compounds are displacing the cap. They are displacing the ClpX. But what's interesting is that the ClpX becomes a target for the activated ClpP. That's something we are interested in, and currently we see it disappearing in the cells. Mm-hmm. But in a test tube, we don't see ClpX disappearing when we activate ClpP. Okay. So we think that we're missing something. We're still not sure what is that we are missing. We're trying to figure it out.
Okay. May I ask one more question? So then, what do you think is? Do you have a handle on what the recognition sequences are in the targets of ClpP that would presumably be recognized by ClpX? Because obviously with the proteasome, there's the ubiquitination system, and we understand that targeting process. But I haven't really been able to find much information on what it is that tells the ClpX to select those particular targets for degradation. And I don't know if you've any idea.
Yeah, I mean, for the ClpXP, for the bacterial ClpXP, we know what is the degron being recognized. This is the SSRA tag, as you know. For the human ClpXP, we do not know.
Okay.
So we have substrates. We have a paper under review. So we know some of the substrates for the human ClpXP, and we know. So we have a list of them, but whether there's a general degron like the case for the bacteria, like the SSRA tag in the mammalian mitochondria, we don't know that at this stage. Okay. And that's something many people are looking for, not just us, So what is a degron in the mitochondrial context,
Okay, thanks, Walid.
Yeah.
Okay, I think we've got time for one or two more questions. Mahmoud, would you like to unmute and ask your question live? Okay. What about compounds' selective toxicity in normal cells compared to cancer cells?
Yeah, so I always get that question. So, yeah, I mean, early on when we started this work, again, I was saying this is going to be just a toxic compound. It's going to kill everything and anything, because basically you're dysregulating ClpP, and there's no difference between a normal cell and the cancer cell. But it turns out that's not the case. The cancer cells are much, much more sensitive than a normal cell, at least in the context of a human body or a mouse body. And now we know for the human as well, they are not toxic. So it turns out that the cancer cells are very heavily dependent on the ClpXP. Its levels are much higher. And therefore, we have the dynamic range needed where you can kill the cancer cell and not really affect the normal cell as much. So we're fortunate that way. And the other thing about the recent approval for the ONC201 is that these compounds can cross the blood-brain barrier, which is really a huge achievement. And that's why they were approved for glioblastomas, because there's really no cure for those types of cancers. So it was, in a way, you can think of it as a low-hanging fruit, a cancer that has no treatment. And this is the first compound that seems to affect it. And therefore, the FDA gave it a quick approval.
Okay, thank you. Are there any other questions? Conrad, follow-up question from Conrad. Go for it.
Hi, Walid. Yes, I have a question regarding the substrates for the activated ClpP. So based on the structures and the amino acid sequence of the substrates that you've now identified, have you been able to, or are you planning to create a type of a model to know which would be the best substrates for ClpP, or the inverse, which would be bad substrates for ClpP going forward, and maybe for drug target identification and validation?
Yeah, so basically, as I mentioned, these are proteins, because we have the sequences and we have the structures, of course, or predict the structures. And so we know that, as I mentioned, it's basically loop regions that are being targeted by the ClpP. Now, in terms of sequences, so we worked very hard to try to see whether there's some general sequence that the ClpP likes to recognize. And statistically, I would say no, and we say no in the paper. But just as an observation, it seems we always see an arginine somewhere close to the cut site. So maybe you need that somehow, but statistically it doesn't pan out, although just based on an observation type thing, it seems that you need an arginine near the cut site. Yeah.
Okay, we're coming up to the hour. If there are any last burning questions, now's your moment. All right. I think, I'm just really struck by how wonderful this webinar series is about bringing colleagues together and getting this conversation going. So I want to say thank you to all the participants who join us for all these sessions and for all your questions at the end. That it really, really spurs us on in the work that we're doing. I've put some links in the chat for your awareness and for your engagement. The H3D Foundation webinar feedback form is a way that you can suggest new speakers and give our speakers some kudos if you want to. I do share any messages that are left there with the speakers. And then obviously a huge thank you to Walid. Thank you so much for carving out the time for us. This really does go a long way to giving back to what we're doing on the continent. So as a member of the diaspora, which I wasn't aware of, but as a member of the diaspora, you can know that you're giving back to us back here on the continent. And we hope that we can loop you into many more of our programs. So just be warned that you'll be getting emails from us in the future. So thank you so much for joining us today and for giving us all your knowledge.
Thank you very much, Nicola, and thank you everyone for taking the time to attend.